This is Unit 22, Digestive System, Part D, the second part. Second part of metabolism includes nutrient pools, nutrient states, blood glucose and its control, regulation of feeding, and body temperature. Now, let's look at nutrient pools. Nutrient pools refer to the total free amino acids, carbohydrates, which amounts to 100% glucose, and fats. Now, these are not located in some tank somewhere in the body, as the term implies, but these are nutrients that are in transport within the body or waiting for transport. They would be in the blood, in the interstitial fluid, and in the other extracellular fluids. Together, they amount to the nutrient pools. From the nutrient pools, nutrients may be put into storage. Carbohydrates and fats, for example, would be stored as adipose tissue, stored in the liver, and in the skeletal muscles. The nutrient pools may be used for energy. Energy would generate a waste product, carbon dioxide. They may be used for building material, cells and molecules used to build tissues and organs. Some is excreted, not a lot, but during the cells and molecules that were produced, maybe lost cells and products that are secreted. Some may be excreted from the nutrient pools, but this doesn't amount to much. The state of the nutrient pools is influenced by the timing of ingestion. Let's look, therefore, at nutrient state. Now, the source of nutrients in the pools is different, depending on one of two states. One is the absorptive state. This is the period of time when nutrients are in the GI tract and being absorbed. It occurs roughly during and after a meal, when nutrients are present in the GI tract. It lasts roughly 12 hours. Post-absorptive state is the other. In this state, nutrients are available from the storage sites. It occurs roughly between meals when nutrients are not present in the GI tract. It lasts roughly 12 hours. Now, these 12-hour intervals are highly artificial. I would say that in this country, because we're fairly affluent, we spend maybe 23 hours a day in the absorptive state because we're always nibbling food. There's always some in our GI tract. But as a frame of reference, we'll use 12 hours for each. The nutrient states involve, as we said, the absorptive state. In the absorptive state, glucose is used as the chief source of energy by all tissues. Excess nutrients, as we said, are put into storage. The glucose is stored as glycogen. 1/4 of the glycogen would be in the liver, roughly 75 to 100 grams. 3/4 would be in the muscles, roughly 300 to 400 grams. And fats. Glucose changed to fatty acids and stored as fats make up another form of nutrients. Fats make up roughly 10 to 30% of body weight. The process is duplicated by insulin. We'll talk about insulin shortly. Fats would be stored in the adipose tissue because they don't need to be used for energy in the absorptive state because enough glucose is available. Proteins, on the other hand, will be used as building material. Any excess proteins, rather than being stored, will be deaminated, that is, removal of the amino group from the amino acids. The amino group components are excreted. The rest is recycled. Because remember, the body has no way of storing excess proteins. What about the post-absorptive state? In this state, nutrients are being removed from storage. Remember, we call that mobilization. Glucose is removed from liver glycogen to supply the nervous system. Fatty acids from adipose tissue supply energy for most of the body's tissues. This is called glucose sparing because glucose is being spared or saved for the nervous system. Caffeine, by the way, increases fatty acid mobilization. If needed, proteins may supply amino acids that are transformed into glucose to be used for energy. This only occurs under very extreme conditions. The amino acids are derived from muscle tissue and from immune system tissue because the muscles contain a lot of protein and the immune system has rapid turnover of protein. So the amino acids are easy to acquire. This occurs under, as I mentioned, very extreme conditions. You may have seen pictures of people recently released from a concentration camp. They look like walking skeletons because their muscle tissue has deteriorated. Many are very sickly for a long time afterward because the immune system has been affected. Starvation promotes muscle breakdown. A crash diet simulates starvation. Muscle and fat is burned, and later hunger is stimulated. Unfortunately, much of the increased calorie intake goes to fat. Let's look at the control of blood glucose. This is a schematic view showing the cardiovascular system as the donut in the middle. Also shown are the livers, the digestive system, the pancreas, skeletal muscles, fat, and other tissues. In the pancreas, two types of cells that we're dealing with are the alpha cells and the beta cells. These cells have insulin receptors, so these cells can monitor blood glucose levels. Glucose enters the blood from the digestive tract and the liver. The normal state of glucose, it serves to supply energy, the major source of energy under normal circumstances. But if it's unavailable, the fats produce fatty acids, which supply the energy. The excess glucose would be excreted. Control of blood glucose levels involves insulin that decreases blood glucose levels, glucagon that increases blood glucose levels, sympathetic nervous system that increases glucose levels. Glycemia refers to glucose in the blood. Normal blood level is roughly 80 to 110 milligrams per 100 milliliters. A1C is another measure. It uses a 3-month average of the glucose. It's based on the binding of glucose to hemoglobin. The more glucose in the blood, the more is bound to hemoglobin. The number measures the percentage of red blood cells that have glucose bound to hemoglobin. Normal is below 5.7%. Prediabetes would be 5.7 to 6.4%. Diabetes, 6.5% or above. If the A1C is 7, blood glucose level is estimated to be around 154. If the A1C is 8, blood glucose levels are around 183. 9, the levels would be 212. 10, the blood glucose would be estimated to be 240. Let's look at control with insulin. The mechanism of blood glucose control by insulin involves glucose being produced by the liver from glycogen. It increases blood glucose level in the blood, and this leads to increased insulin release from the beta cells of the pancreas. The beta cells, remember, have glucose receptors, and as the glucose levels in the blood rise, secretion of insulin also increases. Insulin increases removal of glucose from the blood. This results in decreased blood glucose levels back to normal. Insulin is removed from the blood by the liver in about 10 minutes. Decreased blood glucose would have the opposite effect. We'll return to the role of insulin shortly, as it's quite a bit more complex. What about control with glucagon? The mechanism of blood glucose control by glucagon involves detecting a decreased blood glucose levels, which leads to glucagon release from the alpha cells of the pancreas, and this results in increased glucose release from the liver. It results in increased blood glucose levels, restoring those levels back to normal. Increased blood glucose has the opposite effect. How about the sympathetic nervous system? Mechanism of blood glucose control by the sympathetic nervous system involves a decrease in blood glucose that's sensed by the hypothalamus. The sympathetic nervous system output increases, glucose release from the liver is stimulated by the sympathetic nervous system, and blood levels of glucose rises. Increased blood glucose would have the opposite effect. Remember, in response to stress, the sympathetic nervous system promotes glucose production and secretion by the liver to provide energy to cope with stress. Let's look back at the function of insulin in a little more detail. Function is complex. Its summary is to promote uptake of glucose by tissues and the use of glucose by tissues. But this is, again, quite a simplification. Glucose enters cells by facilitated diffusion. It involves glucose transport proteins. Many types of glucose transporters exist. The types regulated by insulin are mainly found in skeletal muscle and adipose tissue. The following figures will show muscle, liver, and adipose cells. Like most hormones, insulin does more than one thing. That is, it does more than simply transport glucose across a membrane. It mediates different metabolic reactions inside the target cells that create a concentration gradient to allow glucose to diffuse into the cell. Here's an introduction to the function of insulin. The diagram shows a cell. This happens to be a muscle cell. You'll see arrows in the cell. Arrows represent enzyme activity. That may include transport into or out of the cell. If it's a heavy arrow, that indicates a process that's stimulated by insulin. And if it's a dashed arrow, that indicates a process inhibited by insulin. Insulin action will be described in muscle, liver, and adipose cells. Let's look at muscle cells. Glucose is transported into the cell, but insulin also transports amino acids into the cell. Insulin increases protein synthesis and the energy generation from glucose. It also promotes the transformation of glucose to glycogen for storage. In liver cells, it's a little different. Glucose will diffuse into the liver cells from the blood, but only because of a concentration gradient difference. Insulin, on the other hand, promotes the transformation of glucose to glucose 6-phosphate. That continually removes the glucose, maintaining the concentration gradient. It increases cellular respiration. It promotes glycogen storage and increases the production of fatty acids. In adipose cells, insulin promotes the uptake of glucose. It increases fatty acid production from the glucose. Hypoglycemia is a lowered blood glucose level. It may be a decrease in the 20 to 50 milligrams per 100 milliliter range. To restore normal blood glucose levels, a response includes increased sympathetic nervous system activity. Now that leads to increased muscle contractions, shaky feeling, increased sweating. One cause would be a sugary snack that may increase blood sugar levels rapidly. It increases the release of insulin, which decreases the sugar levels quickly, lower than normal, and is hypoglycemia. Insulin shock is a decreased blood sugar resulting from insulin overdose. Hypoglycemia is potentially very serious. It can kill because brain cannot live without glucose. It affects higher brain centers first and then lower brain centers. The symptoms might go roughly like this. The cerebrum would be affected first, leading to shakiness and sweating. The midbrain then is affected. Tonic spasms might occur. The cerebellum is affected. Spasms of extensor and flexor muscles occur. And then the medulla is affected. And this would involve changes in heart rate, nausea, vomiting, decreased blood pressure. Hyperglycemia refers to increased glucose levels. A common form is diabetes mellitus. This results from increased sugar levels in the blood due to a decreased removal from the blood. Type 1 diabetes mellitus, also referred to as insulin-dependent diabetes mellitus, IDDM, or juvenile onset diabetes mellitus. This must be treated with insulin. That is, it depends on insulin for treatment, hence the name insulin dependent. It may occur as late as 25 years of age. 5 to 10% of the diabetics in the U.S. have this form of diabetes. It's almost always found among people with a lean body build. Beta cells fail to produce insulin. This is due to unknown causes, possibly an autoimmune disease. The immune system attacks the beta cells. Viral infection might cause this. Overload of the beta cells during a growth spurt in teenagers might contribute to this form. It's more severe than type 2, which we'll address in a moment. Type 2 diabetes mellitus is referred to as non-insulin dependent diabetes mellitus, NIDDM, or insulin independent, sometimes referred to as adult onset. It may be treated without insulin, so it does not necessarily depend on insulin for treatment. Usually it's diagnosed after the age of 40. 90 to 95% of the diabetics in the U.S. have this form. It includes an obese form and a non-obese form. Obesity is an important predisposing factor. Possible causes include insulin resistance. For some reason, the target tissues fail to respond to the insulin. Beta cells produce less insulin. This might be another cause. Overload of the beta cells in obese adults sometimes results in breakdown of the beta cells. This is less severe than the type 1 form. Another type of diabetes is gestational diabetes. This develops during pregnancy, usually disappears at the end of pregnancy. Other forms are associated with growth spurts, pancreatic disease, malnourishment, and drug use. What about the symptoms of diabetes? We can understand them if we compare the fate of glucose in a normal individual and in a condition with little or no insulin secretion. Under normal conditions, the blood glucose levels are used in cellular metabolism and stored in the liver as glycogen. With little or no insulin, blood glucose cannot be used in cellular metabolism, or it can't be stored in the liver. So what happens? The excess glucose is excreted. In addition to that, because little glucose is available for cellular metabolism, fats supply the energy for metabolism. So there's a decrease of glucose production and an increase in fat production. What about those symptoms? Well, let's look at those that result from an increase in blood glucose. This will increase sugar filtration into the kidneys. The increase in blood glucose then results in sugar found in the urine. The excess sugar in the tubules increases tubular osmotic pressure, which counteracts the reabsorption of water and leads to an increase in urination because if the water is not reabsorbed, remember, it passes out of the body. Because the diabetic is losing water through urination, another symptom is increased thirst. The increased sugar levels in the urinary tract provide nutrients for bacteria, and it often results in an increase in urinary tract infections. Since carbohydrate metabolism has been decreased, one of the signs would be loss of weight. Decreased leukocyte ability to fight bacteria also occurs when an increase in blood glucose results. Causes are not completely understood, but this does lead to more serious infections in general. What about the increase in lipid metabolism? Well, when that happens, there's an increase in lipid metabolites in the blood. These include organic acids and ketone bodies. Acidosis recurs, and the symptoms would include nausea, abdominal pain, headache, and weakness. These ketone bodies and other substances would diffuse into the alveoli, particularly the ketone bodies, because they're rather volatile. This leads to shortness of breath and a strange, sweet-smelling breath. Another consequence of increased lipid presence in the blood would be atherosclerosis. This blocks blood vessels, interferes with blood flow, and leads to poor circulation in the eyes and limbs, as well as elsewhere. It results in gangrene. The gangrene causes blindness and leads to amputation. Increased hunger is referred to as polyphagia. Diabetes was difficult to treat before insulin became available in 1922. Since cells could not use carbohydrates for energy, they depended on fat and protein. The person would rapidly lose weight and at the same time produce increasing volumes of urine. In ancient times, it was thought that the flesh melted into urine. The breath had an odd, sweet smell, and the person suffered from pain and hunger. The person became increasingly weak. Eventually, the person became unconscious and died. The feeding center in the hypothalamus stimulates the desire to eat. A satiety center, also in the hypothalamus, is involved in being satisfied. It decreases the activity of the feeding center, therefore it decreases the desire to eat. The satiety center apparently receives signals from various sources. One is psychological. Another is alimentary, that is the alimentary canal. Stretch of the stomach, for example, tends to increase the desire to eat. Then there are nutritional signals. There are three prominent theories about this. They tend to decrease the desire to eat. For example, the glucostatic theory, increased glucose. Now, we know that if a child eats a candy bar before dinner, it seems to have an effect on decreasing their appetite. The glucostatic signal may work through insulin. Evidence indicates that if food intake is slow, the desire to eat is less. If food intake is rapid, the desire to eat is greater. The lipostatic theory says that increased fat release from storage will decrease the desire to eat. This may work through a signal called leptin. A thermostatic theory says increased body temperature decreases the desire to eat. Let's look at the signals. Short-term signals include ghrelin. Its action is to increase hunger. It seems to have its main action at the start of a meal. It's produced by the stomach, the fundus part of the stomach, by parietal cells in the stomach when the stomach is empty. The target is the hypothalamus. Then there are long-term signals. They include leptin. Leptin's action is to decrease hunger. It appears to be a pretty strong effect. It informs the brain about the number of adipose cells. It's produced by adipose cells, and the target is the brain, presumably the hypothalamus. Insulin has an action to decrease hunger, but this is a rather weak effect. It's produced, as you know, by the beta cells of the pancreas. The target, again, is the brain, presumably the hypothalamus. It may act through another signal called serotonin. Other signals, as I mentioned, include serotonin. Its action is to decrease hunger. It's a rather weak effect by decreasing the activity of the feeding center. It's made from tryptophan. Tryptophan levels are increased by insulin. Insulin released in response to glucose. Therefore, a high carbohydrate diet may decrease the desire to eat. So this would be another mechanism by which a sugary snack before a meal has an effect of decreasing the desire to eat. Low carbohydrate diet may increase the desire to eat. This might explain why dieting often fails. Well, finally, we come to the regulation of body temperature. This involves gain or loss of heat. Before we get to the human, let's talk about the transfer of heat in any object. It's transferred in three ways. Radiation. This is the release of electromagnetic radiation. Conduction. This is the transfer of heat through material, that is, from molecule to molecule. Water conducts heat 20 times faster than air. Water also can absorb much more heat than air. Therefore, wet clothes result in increased loss of body heat. Convection is the third form. It's the transfer of heat by the movement of molecules. Now, these three forms of heat transfer are easily illustrated. Pretend you have a campfire. Fire is burning briskly. You're burning the logs. You stand in front of the fire with your hands up. Your hands get warm by the electromagnetic radiation, the radiation from the fire. What if you, on the other hand, hold your hand above the fire? This would be heating by convection. It's quite hot. The movement of molecules from the flames rise up to your hand. If, on the other hand, let's say you take an iron rod or a fork that has a hot dog on the end of it, you stick that in the fire. If the iron rod or the fork gets hot, the heat is transferred to your hand, molecule to molecule. Heat may be transferred in humans by all three means. Heat gain in humans occurs by shivering, increased metabolism. This is called non-shivering thermogenesis. It's especially common in a type of fat called brown fat. This is found in hibernating mammals and in newborn mammals. Heat loss in humans would be due to water evaporation. Now, when water changes from a liquid to a gaseous form, it absorbs energy to do so. The energy is absorbed from the body. So sweat dripping off the body is less efficient for cooling than simply allowing it to evaporate. Insensible water loss is less important. This is about 1,000 milliliters a day. This is water that's lost through the skin, 400 milliliters, and the lungs, 600 milliliters. It's largely under no physiological control. Sweating causes heat loss. Generally, sweating amounts to 700 to 1,500 milliliters an hour, but it may increase up to 4 liters an hour. Changes in temperatures outside of the body may be detected by thermoreceptors detecting the changes. These are located in the skin. In addition to that, thermoreceptors inside the body detect temperature changes and transmit the information to the hypothalamus. The temperature control center, that is, a thermostat, is in the hypothalamus. It receives the input from the internal thermoreceptors. The center then transmits output to heat-losing centers and heat-promoting centers, and then they transmit information to other areas of the hypothalamus. Various other areas in the hypothalamus send signals to various organs and structures involved in temperature regulation. For example, they send signals to the skeletal muscles, to the hair, to the arterioles in the skin and sweat glands, the adrenal medulla, and the adenohypophysis. Thermoreceptors in the skin also transmit information to the hypothalamus, not shown in the diagram, but they send signals as well to the cerebral cortex. These centers then signal the skeletal muscles to carry out voluntary activities, such as rubbing your hands together or putting on your jacket. Let's look at control if the temperature increases and if the temperature decreases. If the temperature increases, heat is removed by dilating vessels to radiate the heat, by sweating to absorb heat when the liquid water is changed to water vapor. At the same time, those processes that generate or conserve heat are decreased. For example, there would be a decrease in the movement of your skeletal muscles, a decrease in the erection of the hairs. You'll see why that's an effect in a moment. And a decrease in the metabolic rate. Remember, metabolism generates heat. In response, the temperature is decreased, that is, restored back to normal. If the temperature decreases, heat is produced by increasing shivering to generate heat, increasing the rate of metabolism. This metabolism increase is stimulated by two hormones, epinephrine from the adrenal medulla and thyroid hormones from the thyroid gland. Thyroid, in turn, is stimulated by thyroid-stimulating hormone from the adenohypophysis. Heat is conserved by an increase in the erection of hairs. What happens is, as they become erect, they trap air, and the air makes an insulating layer over the skin, so that helps to conserve heat. Constricting blood vessels in the skin also conserves heat because blood transports heat, and with constricting in the skin, there's less heat close to the surface of the body. At the same time, those processes that remove heat are decreased. For example, there would be a decrease in sweating. In response, the temperature is increased back to normal to maintain homeostasis. Let's look at fever. Pyrogens are released from white blood cells to elevate body temperature. Prostaglandins mediate the effect of pyrogens. Aspirin decreases synthesis of prostaglandins, and so the effect of the pyrogens to raise temperature is lessened, and that's why we give aspirin to lower a fever. Hypothalamic thermostat is reset to a higher point when a fever starts. Body temperature at that time is below the new set temperature, so temperature is raised by shivering and by vasoconstriction. Vasoconstriction, as I mentioned, decreases the blood flow through the vessels under the skin, and so the person turns pale. So we talk about shivering, chills, and paleness at the start of a fever. When the thermostat at the end of the fever is reset back to normal, body temperature at that time would be above the new setting, the normal setting. And so the temperature must be lowered, that is, the fever breaks, by sweating and vasodilation. Abnormalities of temperature regulation when temperature increases include heat exhaustion or heat prostration. The thermostat under these conditions functions normally to try to lower the temperature by excessive sweating and by vasodilation. The result, though, may be a decreased blood pressure. This causes a decreased circulation and leads to circulatory collapse. It can be characterized by a cool, clammy skin. Heat stroke or sunstroke, a more serious condition, in this case, the thermostat stops working to try to lower the temperature. So sweating is absent. The body temperature increases. The results may lead to death. And that is the end of the course.